My name is Dilan Karim, and I work here at the university. And I just calculated that for almost more than a third of my whole lifespan, I was a citizen of Semmelweis University, and I still work here. And my vision is that science one day may overcome quackery. I have different ongoing topics, out of which lately I was focused on microbiome research. In this first umbrella project, let's say, we were investigating dysbiosis in the critically ill. The problem during critical illness is that the disease severity itself and the interventions that we apply lead to extreme dysbiosis, which is related to mortality and morbidity. And we still don't know the exact factors that are affecting diversity and to what extent diversity is lost. So our aim was to analyze this. And our hypothesis was that it's very frequent and there are influential factors that we can measure. So we pooled a lot of data and we collected a huge amount of papers together. And after an exhaustive selection, our results came back. And we did a lot of analysis on this huge data. And partially we were happy because we created the largest database ever on the topic, but we couldn't unsee the issues of comparability. On this first analysis, I can say that we were measuring not the real outcomes, but the results represented mostly methodological differences. Let's understand this problem better on this visualization of temporal data. Here you can see different studies assessing the change in diversity over time. And I want to highlight here that there are, of course, changes in diversity over time, but the differences between paper are just much bigger, and that's a huge problem. They are measuring on a different scale, and I have to tell you that most of previous meta-analyses were not so sensitive about pooling this data together. And also the factors we were looking for that are affecting diversity couldn't be examined on this pool microbiome data. We couldn't see any significant interactions with antibiotic therapy, for example, because, I mean, of course, it is there, but the data is not good enough to test it. So on the way to improve comparability, we went further and we dismissed all the papers where we didn't have a healthy control population in the study, and we calculated a standardized mean difference in each study from the healthy control population to eliminate these scale differences. But of course the eligible of studies dropped quickly. But based on this analysis, we could say confidently that microbial diversity is significantly reduced in the critically ill. On this forest plot, trending towards the left means reduced diversity in the critically ill, and there was no difference in the extent of diversity loss among adults and children. On other diversity indices, we got similar results. The richness was also significantly reduced in the critically ill. So a standardized comparison was more reliable, but came with trade-offs. But we need a large analysis which is well-powered, but still reliable results to answer our original questions. So we obtained raw sequence data from online data repositories and from the authors, and our plan is to reanalyze them uniformly and build an individual participant data database, and with this uniformly analyzed data we can work independently from the original paper and the original paper biases. This IPD meta-analysis is considered to be the highest quality of evidence according to Cochrane. We are still building our bioinformatics algorithm and combining it, planning to combine it with free-to-use pipelines. Meanwhile, to further support our research, we collected samples from our own ICU patients for metagenomic sequencing, stool, blood, and respiratory specimens. So now we have our own data from our own ICU sequenced. Based on our way assessing microbial diversity, we want to suggest against using a raw comparison of extracted data and to advise to everyone to compare raw sequence data when working with microbiome. and I call it a Standard Microbiome Research Framework Initiative. It's a very fancy name, and we want to publish this soon. And I had the opportunity to feature this initiative this year at the Colors of Sepsis conference in Ostrava. When I was taking samples from the ICU patients, I was unsure if I'm using the right technique or my different sampling technique makes any difference or not, and data was scarce on the literature on this, so I decided to run a systematic review and meta-analysis to compare sampling techniques, and I found no difference. Also with pneumonia patients, for example, the illness itself and the related mortality is very important, and it's a hot topic, and to have a diagnosis and management, we need good quality samples. To obtain these samples, you can either collect a bronchoalveolar lavage sample, which is very invasive, and it's not very, but it's invasive and resource intensive, but there are less invasive, easier to perform tracheal sampling methods. And once we have a sample, we usually perform culture-based techniques to identify that one or two living virulent culturable bacteria. But we have, on the other hand, this novel emerging technique, metagenomic sequencing, to get a whole landscape of the microbiome there. The results are very different and good for different purposes. And we know that many studies compared sampling techniques based on culturing results, but despite the growing potential of metagenomic sequencing, it was not compared for sampling techniques. So our aim was to perform this. And our hypothesis was that there is no difference between the sampling techniques when analyzed with metagenomic sequencing. So our hypothetical forest plot was anticipated to look like something like this. Diversity is not higher with either method. And of course, if that's true, then it's okay. It's encouraged to go with the safer and more convenient method. So after collecting the 7 eligible articles that we found, those were articles with parallel measurement data, which we really needed for a good comparison. We supported our initial hypothesis, and the results supported the hypothesis. There was no difference between the sampling techniques when used this technique. If you remember, the hypothetical forest plot is very similar to the one that we got. And I want to highlight here the heterogeneity, the low heterogeneity of the first group. I hate this word heterogeneity because it's consumed that much. And if we see a 0% I-square somewhere, it's usually, let's say, we can be sure that it's because of the low amount of included studies. But this is a low amount of included studies, but here I'm quite sure that we are measuring similar populations in this regard, and the similar results of individual studies are causing the low heterogeneity, because these populations were all critically ill ICU patients on ventilator. That is a strength, but with other diversity indices we got similar results. There was no difference between the sampling methods when measuring richness or diversity. So we advise against performing a bronchoalveolar lavage. I want to highlight my work with the risk of bias analysis, because I really felt that choosing an adapted and a very relevant tool has an add-on value only when we are performing these analyses. So for the first project, I built an automated version, an adapted automated version of the NIH quality assessment tool. And for the second project, where we are comparing samples which are obtained from the same patients but via different methods, the traditional items in risk of bias methods were mostly irrelevant. We here want to ensure that the quality of sample is similar in each study. So I conducted a literature review and defined the most important parameters of sources of bias based on that. Did a checklist and sorted items in 4 domains and 2 super domains. And the results of the built analysis is catching very nicely the source of risk of bias. The manuscript is submitted for the 3rd time recently to Scientific Reports. And now that I am about to finish this project, we'll have more time to our ICU related and microbiology related other projects which are also very interesting. And possibly this autumn I will have the opportunity for other international conferences to show my results. Thank you for your attention.
Thank you very much for this presentation, for keeping your time. It's a very modern and interesting concept of microbiome involved in different pathology, even in heart failure, for instance. So now the presentation is open for discussion, please.
Congratulations. Microbiome is a really hot topic nowadays. So I can't read your outcome on the slide, but you examined the length of hospitalization or the first topic. Or what do you see? It's the outcomes is depends on just the Shannon index or the age of the patients and other illnesses like smoking, alcoholic patients, and has any problem examine the subgroups or just see the Shannon index?
Somehow we to examine all those factors that might influence the extent of dysbiosis. We a bit flipped the outcome and the exposure factor. So dysbiosis was an outcome measured by the diversity indices, but we collected all the associated clinical conditions, like as you said, smoking. Smoking was not reported with the patients on ventilator, but high fiber intake, low fiber intake, enteral feeding, parenteral feeding, antibiotic use. We collected all these data plus the severity of illness because we think that they are the most relevant factors. And the outcome was the dysbiosis measures, like the diversity indices, alpha and beta diversity indices we planned. So it's in the the exposure.
And what do you think dysbiosis depends on the problem, or it depends on other parameters like older people's? I think the Shannon index is not the same in the healthy younger patient and on other.
Yeah, the problem nowadays is that to have a good quality evidence, to make a decision for myself on this question, I would need a good quality meta-analysis where lots of studies, or a review where lots of studies results are put together. But unfortunately, this is not possible due to the huge methodological differences now in the technology. There is a publication where a researcher sent her own poop samples to different companies and got vastly different microbiologic results. So what is happening now is that everyone is building a somewhat different analysis pipeline and having results on that and making a model on his or her own sample and saying that age is affecting dysbiosis this much. But it will not be true on the other, and they are not poolable and comparable. That's why we need a standardized research framework, and that's the result that we want to publish, our struggle with answering these questions.
Can I also ask one question? Like, first of all, congratulations, because I think this is truly research, where you don't want to publish ASAP something just to have a publication, but really think about it in detail. So I really admire that. And I would have kind of two questions. So also many diversity indexes you showed us. Do you think some of them is like better or superior or giving more information? And the second, because you show like culture PCR versus these metagenomic sequences. And to be honest, I don't really know how can I use these metagenomic sequences in practice. So I would like to ask this. Thank you.
Thank you. It's very hard to describe dysbiosis. What we can say is that there is a diversity of organisms in any body place, and they are quite diverse. There are a high amount of different species, and they are evenly distributed. That's what we call diversity, and if this diversity is lost or a drastic change happens to the composition of the microbes, then that is dysbiosis. To measure diversity, there are these different indices, and all of them are focusing on something different. For example, Shannon index takes into account the evenness, so how well all the microbes are distributed, and also how frequent, how many different species we can find. But for example, the KO1 index measures mainly the richness, so how many different species can you find it, and it's not very sensitive for evenness. And the Simpson index, I wrote this word like 1 million times down, but it fell out of my head. So the Simpson index is sensitive for evenness more, less than richness. So that's the difference between the indices. And mostly, I would say, for research, this technique is adaptable, and until we can say anything in confidence with this technique, I wouldn't really use it widespread in clinical practice. But in our ICU, when I took samples, for example, there was a patient who had a severe pneumonia. He was resuscitated at the ER. We took different samples for culturing for multiplex PCR, and there was no pathogen found, and the patient got well, got better on the empiric antibiotic therapy, and fortunately he went home. And 1 year after, when I got the results from our study samples, I saw that like 80% of his microbiome was dominated by a Fusobacter species, which is not very hard to culture and very hard to identify. So in this regard, there can be an advance of the metagenomic sequencing.
Thank you. Well, yesterday I was chairing the session, and one of the PhD students presented a work on these advanced molecular technologies in microbiology diagnostics, and compared it to the cultures. And in fact, if I remember well, the performance of these advanced molecular techniques like the PCR and that kind of stuff had a higher false positive event rate as compared to microbiology. Because you are very well in that literature. So it's not really related to this topic, but what is your opinion on the routine PCR sampling? Because I think that could be an important addition to your findings that doing a regular routine bronchoscopy is a waste of time and effort, and doing PCR testing. So what is your opinion and comment on that?
Yeah, I didn't really want to touch this topic, and that's because there were really many studies arguing for decades on this, whether to perform a lavage or a simple tracheal sampling, and the results are quite mixed. And the metagenomic sequencing is not very good. It doesn't, in a regard to identify a pathogen, it doesn't have a good sensitivity, because it's just identifying every living, dead DNA which is there. In this regard, the multiplex PCRs are a bit better, because they are only amplifying 10 or 20 sequences which are directly related to the mostly observed pathogens. But they are very expensive, and in my opinion, they don't really have that much added value, because anyway, they are never coming back in 2 hours, and in half or 1 day, the results are coming back. We already started an empiric antibiotic therapy, and on the next day, the culturing results comes back as well, and it costs 80,000 forints, and the other costs 2,000 forints. And of course, you need culturing as well, because you need sensitivity. What is that? Antibiotic susceptibility. So, but that's a personal belief. yeah, there is debate on the topic regarding to this.
You used very often this term metagenomic sequences.
Oh, yeah.
Yeah. What do you... So how do you explain this terminology, metagenomics? Is a little bit in a
Shady.
sense of, of a philosophical one. We speak very often about metamental, for instance. So,
yeah, so I also looked that up when I was first reading what is this actually doing this technique. So it's called metagenomic because it's collecting a sample, collects all DNA from it, and amplifies only the bacteria DNA. But that's a metagenomic mix of every bacteria that is found there. So that's why it's called metagenomic, because it's on one reason. So on one aspect, it's not the host's genome, it's an additional genome. That's why it's metagenomic, and it's a mixture genetic material which is broadly sequenced and amplified, and then the later analysis will...
I agree with you. And did you think the relationship between genomics and epigenomics? Because speaking about some factors, they could influence this presentation of the patients.
Yes, I told you this technique is sensitive only, but not this is sensitive to living, dead, virulent, non-virulent bacteria. So there is much noise. We don't actually see what genes are expressed in these creatures, but if we are working with RNA, then we are basically working with epigenomics, and it's also possible, but as you know, might know, working with RNA is much more expensive, and it's very sensitive to any temperature changes and sampling handling. So there is the data is very scarce on the epigenomic, but it's possible. Only you have to work with RNAs instead of DNA.
Hello, congratulations on your presentation. It was amazing. And I wanted to ask you about, so all these indices that you measured are basically quantitative, right? Have you thought about measuring any qualitative difference, indices or anything like the different bacteria?
Yeah, of course, that's a very good question, because when you think of a microbiome, you think of bacteria and not their diversity. I mean, for me, that's not catching the core, but unfortunately, you need sort of standardizable, quantifiable measures to do the meta-analysis.
It's the pipelines that you said it's the problem.
No. I mean, for a meta-analysis, data is not reported in a comparable way. For example, stacked bar plots, like 20% of, Fusobacteria, 30% of Archaeobacteria, that is without a measure of spread and the known number of sequenced bacteria is not poolable, so we cannot work in a meta-analysis in that. Maybe we couldn't work, but maybe there is a way, but it's not invented yet to pool these. But in every study, of course, they are focusing on the question that you are asking. So in a study that you are conducting, you are getting a result like this. So on the right side, you can see a stacked bar plot, and you can see different bacteria, types of bacteria, which percent contributes in a relative amount to the whole microbiome, and that's have more information than the diversity.
Congrats on your presentation. I was just wondering whether it would be possible to not compare the exact results to each other, rather the changes in the indices, so maybe a baseline index and then the change through the disease.
Yeah, that was my idea as well, but I had to rush through very fast. So the original results were just collecting and pooling the results of the indices, but you need a comparator to have this comparison, and there is no a widely accepted range, like in healthy controls, Shannon index is 4. You cannot say that. So that's why we dismissed all papers which didn't have a control group, and we worked with the papers where there was a control group and their diversity, healthy control group, and their diversity was measured with the same techniques, with the same pipelines. So we basically did a standardized mean difference, as you said. We just checked in each study what is the difference from baseline in that study, which were the healthy controls.
Thank you.
If you would have this idea like 1 year ago, you would have helped me much better.
2 questions. One is just as simple so that I understood the mode that you did the research correctly. So you first calculated the Shannon indices or the indices, whatever indices, and after that, to remove the so-called heterogeneity, you calculated the standardized mean difference of the indices. So you took the Shannon index and treated it as a continuous variable and calculated the standardized mean difference between the two, the healthy and the...
Between the healthy and the ill, yeah.
Okay.
And with that, the huge variance differences were somewhat eliminated, but still some heterogeneity is included because of pre-processing.
Yeah, of course. And my second question is that you've talked about the frequencies and stuff of different pathogens, but you're talking about relative frequency, which is really, really dependent on the primer you used. So what can you really compare to diversity indices calculated based on 2 different primers?
Here, with metagenomic sequencing, the technology is not using primers, but it's even a bigger problem. So the primer is only accessing the 16S RNA coding DNA, and that is amplified, which is cleaning up all the bacterial stuff there. And then is just cutting everything in the length that is needed for next generation sequencing and just broadly sequencing every result. And then the bioinformatic pipelines are aligning it to known species, which has even more error in it, depending on the database that is aligned to, depending on the aligning mechanism. So there are a lot of steps where there can be little differences which will really add up, and as you say, will contribute to the differences that we observed.
So thanks. So in few words, your aim is to give a protocol, please do this. And
yeah, I mean, maybe just to talk more about this problem, because nowadays you can buy your own gut microbiome analysis, and in Hungary as well, there is an institute. You go there, you pay 300,000 forints, and they will say you to eat more carrots and run twice a week, and you will feel healthier. And then, yeah, but you cannot know based on this data, so more like this.